I will continue with general virology lecture and will discuss laboratory diagnosis of viruses. I have already discussed that viruses are obligate intracellular parasites and they need to have a living cell for their growth. So you can either cultivate these viruses in animals, embryonated eggs, or tissue cultures. Animal inoculation, for example, mice, guinea pigs, monkeys, rabbits, etc. And route of inoculation can be intracerebral, interperitoneal, intranasal, subcutaneous, etc. The animals are used for primary isolation of viruses which do not grow in eggs, these embryonated eggs and tissue cultures, to study the immune response, to study the pathogenesis of viral diseases. But there are certain disadvantages of this method. There can be interference of immunity with viral growth, infection of animal with latent viruses. There is high cost, difficulty in handling, maintenance of animals. So these are the issues with this animal inoculation method. However, if you compare this with embryonated eggs, for example, hen's egg, it's simple to handle, it's cost effective and easily available, and there is no immunological interference with growth. So you can see that there are different routes of inoculation in hen's egg, chorioallantoic membrane, similarly injection into amnion, yolk sac, etc. Tissue cultures, I'll talk about this in next slide. So tissue cultures. Tissue cultures are of three types: organ culture, explant culture, and cell culture. Organ culture means the whole organ is taken. Explant culture, fragments of minced tissue are taken. And in case of cell culture, there is a proper method of forming the monolayers for cell culture. And the desired tissue is cut into small pieces. And this is treated with enzyme, for example, trypsin, and by mechanical shaking, tissue is dissociated into the component cells. These are then washed, counted, suspended in a suitable growth medium containing essential nutrients, for example, amino acids, vitamins, salts, glucose, etc. And it's also supplemented with up to 5% of calf or fetal calf serum. The cell suspension is distributed in a glass or a plastic container, and on incubation, the cells adhere uh, to the glass or plastic surface and divide to form confluent monolayer sheet of cells within a week. So, based on their origin, chromosomal characteristics, and number of generations through which they can be maintained, cell cultures are classified into three types: primary cell cultures, diploid cell cultures, and continuous cell cultures, or you can also call them as cell lines. So let's see the differences between these three cell lines. Primary cell line, these are taken from normal cells, freshly taken from the organs of animals or humans and cultured. However, there is a limitation. It can't be maintained in serial cultures. There is a limited growth, just 5 to 10 divisions, and it is used primarily for virus isolation and their cultivation for vaccine production. The example is of rhesus monkey kidney cell culture. Diploid cell line. These are cells of a single type that retain the original diploid chromosomes and karyotype during serial subcultivation for a limited number of times. These cells are developed from human fibroblasts. They can undergo many more cell divisions than the primary cell cultures before dying out. They can undergo around 50 divisions. They're used for isolation of fastidious pathogens and vaccines production. The example is human embryo lung tissue. Continuous cell lines. These cells are derived from monkeys, cats, dogs, and humans, etc. They are derived from cancerous tissue. These cells grow faster and their chromosomes are haploid. They are capable of indefinite growth in vitro. They are used for antigen production, isolation of viruses from clinical specimens, and vaccine production. The example is human carcinoma of cervix cell line. There are various methods to identify viral growth in cell culture. The first one is to observe cytopathic effect, and this is defined as detectable morphological changes, such as change in size, shape, and fusion of cells to form multinucleated giant cells. So detectable morphological changes in the appearance of a virus-infected cell. It gives you a presumptive identification of a virus growth in cell culture, and at the same time it's a manifestation of virus-infected cells that are dying or dead. Let me give you an example of measles and respiratory syncytial viruses. There is formation of a syncytium, fusion of infected cells with adjacent cells to form giant cells containing several nuclei. Another example is of rounding and aggregation of cells into grape-like clusters. So these are the examples of cytopathic effect. Another method is hemadsorption. And in this case, there is attachment of erythrocytes to the surface of virus-infected cell. This technique is limited to viruses with a hemagglutination protein on their envelope, such as mumps, parainfluenza, and influenza viruses. There is another method which is called as interference. So in this case, non-cytopathogenic virus, for example, the rubella is a non-cytopathogenic virus. So if it is present, it can interfere or inhibit the activity of cytopathogenic virus, which is Coxsackie virus as an example. Decrease in acid production by dying cells. This can be detected visually by a color change in the phenol red, a pH indicator in the culture medium. So the indicator remains red in the presence of virus-infected cells, but turns yellow, acidic, in the presence of metabolizing normal cells as a result of acid production. So this technique can be used to detect enteroviruses. There are certain serological tests for definitive identification of viruses in cell cultures. The first one is hemagglutination inhibition method, and in this method, if viruses and their antibodies are homologous, then these viruses are blocked from attaching to erythrocytes and there is no hemagglutination. So this is called as hemagglutination inhibition method. Obviously, this is possible only for those viruses which can agglutinate RBCs. Then there is another method which is called as neutralization. Here you can see that these viruses can easily infect the cell, but if these viruses and their antibodies are homologous, then these antibodies which have attached to the surface of these viruses will block the entry of viruses into the cell. So this is called as neutralization. Then is complement fixation method. If you remember, I have been talking a lot about complement fixation, ELISA, fluorescent antibody tests in parasitology laboratory diagnosis and also in other laboratory diagnosis of special bacteriology. So today we will discuss these methods. First, the complement fixation. Please remember that complement system consists of 20 or more plasma proteins that can interact with one another and with cell membranes. Each protein component must be activated sequentially under appropriate conditions for the reaction to progress. Antigen antibody complexes are among the activators, and the complement fixation tests can be used to identify one of them if other is known. So this reaction consists of the following two steps: antigen and antibody. One is known and the other is unknown. Example, use a known antigen and determine whether a patient's serum contain antibodies to that antigen are mixed and measured amount of complement. And this is actually usually from guinea pig is added. If antigen and antibody match, they will fix up this complement system. So if both of these are going to match, they would attach to complement system. And once the indicator system, which consists of sensitized red blood cells, that is, red blood cell plus anti-red blood cell antibodies are there in this indicator system, so they are added. So if the antigen antibodies which match each other, they have fixed the complement system, so less complement system is available to attach to this indicator system, and the red blood cells remain unhemolyzed, and this test is positive one. So there is no hemolysis and this is a positive test, and it means that the patient's serum had antibodies to that antigen. In case if antigen and antibodies, they don't match in the first step, so they would not fix the complement, and much of the complement is free to attach to this indicator system, sensitized RBCs, so they are lysed and this would produce hemolysis, and this is a negative test. Enzyme linked immunosorbent assay test, which is a very important test. It measures either antigens or antibodies in patient specimens. For measurement of antibody, known antigens are attached, or you can say they are fixed to the surface. Example, the bottom of a small well on a plastic plate. It is incubated with dilutions of patient serum, and if this antibody is homologous, it is going to attach to this known antigen, and obviously this is the unknown antibody that we have to find out. Then it is washed and then re-incubated with antibody to human IgG labeled with an enzyme which is called as horseradish peroxidase enzyme. Enzyme activity is measured by adding the substrate for the enzyme. This is the substrate. And estimating the color reaction in a spectrophotometer. So the amount of antibody bound will be proportional to the enzyme activity, and the titer of antibody in the patient's serum is the highest dilution of the serum that gives a positive color reaction. Fluorescent antibody test. These are of two types, direct and indirect immunofluorescent test. First, I will discuss this direct immunofluorescent test. In this case, an unknown antigen, for example, from a tissue of a clinical specimen, is fixed on a glass slide, and specific antibody tagged with fluorescent dye is applied directly to the slide. It is allowed to react for 30 to 60 minutes at 37 degrees centigrade and then is observed for fluorescence under fluorescent microscope. So it is made visible by UV light in the fluorescent microscope. In case of indirect immunofluorescent test, as you can see over here, the known antigen is attached to the slide, and if you have to find out the unknown antibody from the patient's serum, so you would add the patient's serum and the homologous unknown antibody would attach to this antigen. The next step is to attach fluorescent dye labeled antibody to human IgG, and then it is examined by ultraviolet microscope. This indirect test is more sensitive than the direct immunofluorescent test because more labeled antibody adheres per antigenic site. This labeled antiglobulin becomes a universal agent, that is, it is independent of nature of antigen used because the antibody to IgG is reactive with all human IgG. So this is indirect immunofluorescent test. Then is radioimmunoassay. This is mostly used for quantitation of antigens or heptins to assay hormones or drugs in serum. And as you can see over here, the first step is to add fixed amount of radiolabeled antigen, which is then mixed with a known amount of antibody for that antigen, and they would bind to each other. The next step is to add a sample from a serum of a patient containing unknown quantity of the same antigen. Once it is added, this causes the unlabeled antigen from the serum to compete with the radiolabeled antigen for the antibody binding sites. The complexes between the antigens and antibody, they can be separated and the amount of radioactivity is then measured. The more unlabeled antigen is present, the less radioactivity would be there in the complex. Then is immunoelectron microscopy. If the antibody is homologous to the virus, so the aggregates of virus antibody complexes can be seen with the help of electron microscope. Microscopic identification with the help of light microscope, and the example would be of multinucleated giant cells to see the cytopathic effect, ultraviolet microscope for fluorescent antibody staining of virus and infected cell, electron microscope to see the size and morphology of virus. Then is detection of viral antigens and viral nucleic acids. So this is direct detection, and for example, in case of hepatitis B surface antigen, this is the antigen which can be detected, and then nucleic acids in case of hepatitis B PCR, hepatitis C PCR, etc. So what is the summary of lab diagnosis of viral infection? So you can either isolate this from cell culture, animal inoculation, embryonic eggs, tissue culture. We have talked about all of these different inoculation methods. You can find out cytopathic effect. There is another method, heme adsorption interference method, and you can also detect viruses in a cell culture with the help of serology, like complement fixation test, hemagglutination inhibition test, neutralization test, ELISA. We have radioimmunoassay, etc. Direct detection with the help of microscopy, antigen detection, and nucleic acid detection. And we have already discussed the examples. This P24 antigen is for HIV virus. Thank you.